everyone. Today our group will be discussing histamine and antihistaminic agents. This topic is important because histamine plays a major role in allergic reactions and other physiological processes. We will also explore how antihistamines work and their medical applications. Histamine is a substance produced by our cells to help the immune system respond to allergic triggers. It is a biological active compound that plays a crucial role in various physiological and pathological processes. Histamine is synthesized from the amino acid histidine by the enzyme histidine carboxylase and is stored mainly in the mast cells, basophils, and some neurons. Antihistamines are drugs that block histamine by preventing it from binding to its receptors. There are two main types of antihistaminic agents. We have H1 receptor antagonists or H1 antihistamines. These are used to treat allergic reactions, motion sickness, and can also have sedative effects. Second, we have H2 receptor antagonists or H2 blockers. These are primarily used to reduce gastric acid secretion and treat conditions like ulcers and GERD. The structure-activity relationship, or SAR, refers to the connection between a molecule's chemical structure and its biological activity. Understanding SAR helps scientists modify drugs to improve their effectiveness, potency, and selectivity, making them more suitable for specific medical conditions. Cetirizine is a second-generation H1 antihistamine, primarily used to treat allergic conditions such as hay fever, urticaria, and skin allergies. It works as a selective antagonist of histamine H1 receptors, meaning it blocks histamine's effect and prevents allergy symptoms. Compared to older antihistamines, it causes less sedation, making it more suitable for daily use. Cetirizine is derived from hydroxyzine, an older antihistamine. However, it has been structurally modified to improve selectivity and reduce central nervous system side effects. These modifications make cetirizine more effective while minimizing drowsiness. Now let's break down the key structural components of cetirizine. First, we have pyrazine ring. This is present in both cetirizine and its parent compound. It plays a crucial role in binding to H1 receptors, ensuring strong antihistamine activity. Next, we have the carboxyl group. This increases polarity and hydrophilicity, which prevents the drug from crossing the blood-brain barrier significantly. As a result, cetirizine causes less sedation. Lastly, we have chlorophenyl and benzyl hydryl groups. This contribute to lipophilicity, allowing cetirizine to bind effectively to histamine receptors. The structure-activity relationship, or SAR, of cetirizine explains why it is highly selective and effective. The piperazine ring ensures strong receptor binding. The carboxyl group prevents the drug from entering the brain, reducing sedation. Lipophilic aromatic rings enhance receptor interaction while maintaining peripheral selectivity. The absence of tertiary amine further minimizes central nervous system effects compared to first-generation antihistamines like diphenhydramine. In summary, cetirizine is a well-designed antihistamine that balances efficacy with safety. Its carboxyl group and increased polarity making it non-sedating, while its piperazine core and lipophilic rings contribute to strong receptor binding. These structural modifications make cetirizine a long-acting, selective, and effective antihistamine with minimal CNS side effects.
What is loratadine? Loratadine is a long-acting second-generation, non-sedating, tricyclic antihistamine with selective antagonistic properties to peripheral histamine H1 receptors. Loratadine selectively inhibits H1 receptors primarily located on respiratory smooth muscle cells, vascular endothelial cells, the gastrointestinal tract, and immune cells. Unlike first-generation antihistamines such as diphenhydramine, loratadine is a competitive histamine antagonist that does not cross the blood-brain barrier. Therefore, it does not affect the neurons of the central nervous system, thereby preventing daytime somnolence or sedation. Uses of loratadine. Loratadine is used to treat allergy symptoms such as allergic rhinitis and urticaria. In allergic rhinitis, loratadine effectively relieves symptoms like sneezing, runny nose, itchy eyes, and itchy throat caused by allergies to pollen, dust, or other airborne substances. And in urticaria, it helps reduce itching and redness associated with hives. However, it doesn't prevent hives or other allergic skin reactions. Additionally, loratadine can be used to treat other skin allergies such as eczema. The structure-activity relationship of loratadine includes benzocycloheptapyridine core, chlorosubstituent at position 8, piperidine ring attached via an exocyclic double bond at 11 position, and an ethoxy carbonyl group on the piperidine ring. Benzocycloheptapyridine core is crucial for maintaining the optimal conformation needed to interact with the H1 receptor. This means that it is the central rigid structure of the loratadine molecule. Benzo indicates a benzene ring fused to a 7-membered ring containing a nitrogen atom or pyridine. The rigidity of this core is crucial. Flexibility in this region would likely disrupt the precise spatial arrangement needed for optimal binding to the H1 receptor. The rigid structure helps maintain the correct orientation of the other functional groups. Next is the chlorosubstitute at position 8. It enhances the binding affinity to the H1 receptor, which means that the chlorine atom at position 8 plays a significant role in enhancing the drug's potency. Chlorine is a relatively small electronegative atom. Its presence likely affects the molecule's electronic distribution, making it better able to interact with specific amino acid residues within the H1 receptor's binding pocket through van der Waals forces and possibly dipole-dipole interactions. The precise position is critical. Changing the position of the chlorine atom would likely reduce or abolish activity. Lastly, the piperidine ring at position 11. It is essential for receptor interaction and contributes to the overall potency of loratadine. The piperidine ring is attached to the benzocycloheptapyridine core at position 11. The exocyclic double bond means the bond connecting the piperidine to the core is outside the main ring system. In the conformation, the double bond restricts rotation around that bond, maintaining a specific 3-dimensional conformation of the piperidine ring relative to the core. This specific conformation is essential for optimal interaction with the H1 receptor. In hydrophobic interactions, the piperidine ring contributes to hydrophobic interactions within the receptor's binding pocket. These interactions help anchor the molecule in place. In steric factors, the size and shape of the piperidine ring as well as its position are critical. Changes to the piperidine ring would likely alter binding affinity. In conclusion, the overall arrangement of the benzocycloheptapyridine core, the chlorosubstituent, and the piperidine ring is crucial for optimal binding. Even small changes in the relative positions of this group could significantly impact the drug's activity. This special arrangement allows for multiple favorable interactions such as hydrogen bonds, van der Waals forces, hydrophobic interactions, with the H1 receptor leading to high binding affinity and potent antagonist activity.
Good day to you all. Now let us proceed to another antihistamine drug, which is diphenhydramine hydrochloride, or is famously known as Benadryl. So, this is an antihistamine that may be used for the treatment of allergic reactions, and this type of drug belongs to the first generation antihistamines and belongs to the ethanolamine class. So diphenhydramine treats allergic type reactions by blocking histamine 1 or the H1 receptors. So this prevents histamines from affecting the airways, blood vessels, and the gastrointestinal tract, reversing symptoms such as bronchoconstriction, rash, and itch. So when histamine binds to the receptor on cells, it stimulates changes within the cells that leads to what I've said before, the allergic reactions. So basically, antihistamines compete with histamine for cell receptors and bind to the receptors without stimulating the cells. So diphenhydramine also blocks the action of acetylcholine, and this causes drowsiness because it has its sedative effects when the drug is taken. So let us proceed to the structure-activity relationship. So before that, the IUPAC name for diphenhydramine hydrochloride is 2-diphenylmethoxy and N-dimethylethanamine. So functional groups present in this structure is diphenylmethane, which is the backbone of the structure, the ether linkage, the ethyl linker, and the tertiary amine group, and the hydrochloride salt ion. So let's dig deeper on the structure-activity relationship of these functional groups. So first, let us start with diphenylmethane backbone. So this consists of 2 aromatic rings, and they basically contribute to the lipophilicity of diphenhydramine. So again, this is essential for its activity, and its structure is very common in many first-generation antihistamines and is thought to be responsible for their H1 receptor antagonism by blocking histamine receptors. So the 2 benzene rings, as I've mentioned, increase the lipophilicity, allowing diphenhydramine to easily cross the blood-brain barrier, or also known as the BBB, and contributes to sedative effects due to CNS penetration. Next, we have the ether, or also called as the ether linkage. So this maintains an optimal distance between the lipophilic rings and the amine group, crucial for receptor binding. The presence of an ether linkage in diphenhydramine contributes to its stability and overall structure. Next, we have the ethyl linker. This is the ethanolamine moiety, provides flexibility for receptor binding. So the ethanolamine moiety is a key pharmacophore in first generation histamine 1 or H1 antihistamines. This 2-carbon distance allows the molecules to flexibly adopt the right conformation to fit into histamine's receptor binding pockets. So if there are any change in the length of the ethyl linker, it can reduce the antihistamine activity. So if it is short, then it can lead to reduced binding affinity because nitrogen is just too close to the rest of the molecule. If it is lengthened, it disrupts the ideal positioning and may reduce antihistamine activity. So the ethyl linkage also enhances lipophilicity, allowing it to cross the BBB efficiently. That is why diphenhydramine not only acts as an antihistamine, but also has strong sedative effects by blocking H1 receptors in the central nervous system. Next, we have the tertiary amine, which is responsible for H1 receptor antagonism. So it blocks, or blocking histamine binding increases the CNS penetration, leading to sedation and drowsiness. Lastly, we have the hydrochloride salts, which enhances water solubility, improving oral absorption and bioavailability. So the pharmacological effects based on the structure-activity relationship of the functional groups in diphenhydramine. So then again, it is an H1 antagonist. It blocks the histamine receptors, and then it reduces the allergy symptoms, which concludes that it has the antihistamine effect. Next is the CNS penetration. So diphenhydramine penetrates the CNS efficiently due to its lipophilic nature. That's why it crosses the BBB efficiently. Its small size and lack of strong hydrogen bonding. So with this, it causes sedation and drowsiness. So to conclude everything that I've said, so diphenhydramine hydrochloride is a first generation H1 antihistamine with sedative and anticholinergic activity. Its lipophilic aromatic rings enable BBB penetration and causes intense sedation. And the ether and ethyl linker, or the linkage, are essential for H1 receptor binding. So the tertiary amine also is responsible for the receptor antagonism, and its hydrochloride salt enhances solubility and bioavailability. So clinically, diphenhydramine finds application in allergic reactions, basically the sneezing, runny nose, and itching, most especially the hay fever, sinusitis. So its CNS depressing effect restricts daytime use, because if you're, for example, driving, then it can, and you're taking Benadryl, it causes you to be drowsy and sleepy, and it can be dangerous when driving. So I think that's all of it for diphenhydramine.
Next is the doxylamine. It is a first-generation antihistamine belonging to the ethanolamine class, widely used as a sedative, anti-allergic agent, and antiemetic. Its pharmacological effects are primarily attributed to its antagonistic action on histamine H1 receptors, along with moderate anticholinergic activity. This is the structure of doxylamine. This part is the ethanolamine backbone. Ethanolamine backbone, it is essential for H1 receptor binding and activity. It enhances water solubility and contributes to antihistaminic properties. The next one is a diphenyl ether system, or the 2-benzene ring. These 2 phenyl rings here increase lipophilicity, allowing better blood-brain barrier penetration and enhancing sedative effects. This part is a pyridine ring. A pyridine ring, or the 6-membered aromatic ring with a nitrogen atom, replaces one of the phenyl rings found in similar antihistamines like diphenylhydramine. This modification alters metabolic stability and receptor affinity, making doxylamine distinct in terms of potency and duration of action. Lastly, the tertiary amine. The presence of a dimethylated nitrogen allows interaction with H1 receptors and contributes to its anticholinergic effects. This amine group plays a role in receptor binding, influencing sedation and antimuscarinic side effects like dry mouth and dizziness. Next is the structural activity relationship and the pharmacological activity of doxylamine. The activity of doxylamine is strongly influenced by its structural features. The first is the H1 receptor antagonism. Doxylamine competes with histamine at H1 receptors, reducing allergic responses and causing sedation. Next is the central nervous system penetration. It is a highly lipophilicity diphenyl moiety enhances penetration into the central nervous system or the CNS, contributing to its sedative properties. Lastly, the muscarinic antagonism. It moderates inhibition of muscarinic acetylcholine receptors, contributes to side effects such as dry mouth, dizziness, and blurred vision. So in conclusion of the doxylamine pharmacological effects are strongly dictated by its structural features. Its diphenyl and ethanolamine moieties contribute to potent H1 receptor antagonism and significant sedation. Modifications targeting muscarinic receptor affinity and lipophilicity can fine-tune its efficacy and side effect profile. So in understanding this structural activity relationship principles aids in the development of improved antihistamine with optimized therapeutic properties.
Hi everyone, in these slides I'm going to tackle about the chemical structure, the functional groups present, the core structure, the implication of the structure-activity relationship of the drug named clemastine. So what is clemastine? Clemastine is an antihistamine drug with a moderate sedative properties. It is used to treat allergic reactions like high fever and common colds. It is also help in relieving symptoms like runny nose, sneezing, itching, watery eyes, and skin allergies like hives. Also, clemastine works by blocking histamine. So what is histamine? Ang histamine is mauna siya ang substance sa atong body nga gi-release during allergic reactions. Clemastine is a first-generation antihistamine, meaning it can cause drowsiness and sometimes gamiton ni siya as a mild sedative. If we say mild sedative, mauni siya ang nag-induce og calmness, relaxation, and drowsiness without causing deep sleep or significant impairment. Now, let's move on to the chemical structure and the functional groups present in the drug clemastine. So there are 5 functional groups present in the clemastine drug. These are the aromatic hydrocarbon, which is the benzene ring attached in the slide. Second is the halogenated aromatic hydrocarbon, which is the chlorobenzene ring group. The benzene ring with the Cl attachment. Next is the methyl group, the CH3 attachment na adere nakabutang. Next is the aliphatic ether, the attachment nga O and then duha ka line. And then the tertiary amin the H and CH3 attachment. Now let's move on to the core structure and the functional groups present in the clemastine drug. So basically this slide is just a summary of the recent slide. And it is where I will explain ug asa sila na-attach ug asa sila nakabutang ug unsay purpose pa pagbutang sa ilahang mga structures. So, the first 2 structure present in the clemastine drug is the aromatic hydrocarbon and halogenated aromatic hydrocarbon. So, clemastine drug contains a central benzihydryl ether structure where one of the phenyl rings is substituted with a chlorine kato Cl nga attachment sa nakabutang sa benzene ring sa ubos. This makes the atom called a chlorobenzene ring. So aromatic hydrocarbon provides a hydrophobic interaction which siya help sa drug nga mo bind sa histamine or H1 receptor sa atong lawas. It enhances lipophilicity, which allows clemastine to cross the BBB or the blood-brain barrier and siya ang makakause og sedation. While the halogenated aromatic hydrocarbon or the chlorobenzene ring group
Naani siya chlorine atom nga nag-increase og lipophilicity, and it helps drug to stay longer in our body by reducing its metabolism. So maka-help ni siya nga mahinay or mukuan ang metabolism sa drug sa atong lawas. It also strengthens the binding to H1 receptors, which can improve the potency of the antihistamine sa ato ang lawas. Second functional group present in the clemastine drug is the aliphatic ether. This refers to the ion ether where the oxygen atom is connected to the two alkyl groups. Moto katong O and then ay duha ka attachment. That's what we call alkyl groups, or the carbon chain without aromatic ring. So basically, ang tawag niya ato is the aliphatic ether. Aliphatic ether or katong attachment nga circle there. Aliphatic ether helps in hydrogen binding, which may assist in receptor binding and solubility. It increases molecular flexibility, allowing the drug to adapt the shape for the receptor interaction. Next functional group present in the clemastine drug is the tertiary amine. So tertiary amine is basically katong methyl nga functional group niya, gi-attachan na siya og nitrogen atom. So that's mao na siyang gitawag siya og tertiary amine. The amine or the HNCH3 attachment. It is the key bonding for H1 receptor as it interacts with receptor site via ionic hydrogen binding. It can affect the CNS activity, contributing the clemastine sedative effect. So maani siya nakahatag og sedative effect sa drug nga clemastine, which is nagkuan siya og katong calmness, relaxation, drowsiness without having deep sleep or any impairment. It also helps in determining the drug basicity, influencing how it interacts with the biological membrane. So basically, these functional groups present in the drug clemastine has antihistaminic effect, sedative effect, and long-acting properties sa drug. Next functional group is the methyl. Methyl is the part of the pyrrolidine ring specifically attached sa nitrogen atom. So katong tertiary amine nga gi-attachan og nitrogen, nahimo siya tertiary amine. So katong methyl mao to siya ang CH3 nga attachment sa clemastine group, which is the CH3 attachment. It slightly increases lipophilicity, which enhances BBB penetration and can help in the central nervous system effects. It can affect drug metabolism by making the molecule more resistant to breakdown by the enzymes. Now, let's move on to the implications of the SAR, or the structure-activity relationship of the clemastine drug. There are two implications ang clemastine drug. It is the modifying the aromatic rings and the altering the side chains. So, in modifying the aromatic ring, it is where the changing of the substituents on the aromatic ring can affect the binding affinity and the overall activity of the drug. So example ani nga kuan kay katong sa hydrocarbon og halogenated hydrocarbon nga giadan og chlorine attachment ang benzene ring. That's why para mahimo siya chlorobenzene ring. The altering the side chain. Modification to the amino alkyl chain, such as changing the length and introducing different functional groups, can impact the drug's potency and potential side effects. So ang example ati kay katong methyl group nga giadan og nitrogen, nahimo siya tertiary amine nga, and the tertiary amine kay mao to siya ang naka-cause og sedative effect sa drug and naka-reduce sa metabolism sa drug and affect siya sa potency and efficacy sa drug nga clemastine. So, in conclusion to this slide, clemastine's primary functional group is the ethanolamine group, which is very crucial to the activity as the first generation antihistamine drug. This drug has two major works, the antihistamine activity and the sedative activity. So in the antihistamine activity, it is where the drug will prevent histamine from activating the receptors and causing allergic reactions. While in the sedative effect, it is the ability to cross the receptor sa BBB or sa blood-brain barrier easily.